but it affects the catalytic activity of the enzyme, or the third concept is that it gets mistargeted and it's taken away from the site where it needs to be, which is the lysosome, away from it. So now if I can prevent all this from happening, hopefully my enzyme will go to where it needs to go and there's going to be a good response. So your scenarios where you can develop an immune response in an autoimmune condition, you're already in a state of producing antibodies. So this is going to be in a setting of following exposure that you're already making it. Or in the setting of an enzyme treatment, we have the advantage, if I can identify the patient at risk, for instance, the cross-reactive immunological material negative, which is a whole different story, we now have a way to do that. Then in the naive setting, I can try and re-educate the immune system and prevent the antibody response. And that's the part I'm going to focus on today. So we looked at this diagram, and the concept is now, if I can use a drug that prevents this whole cascade from occurring, I'm going to prevent an immune response. And please, this is trying to show you how we're making advances. Each one of these, to me, is an advance. And so we developed a protocol. The details are not important, but we used drugs like a B-cell agent, rituximab, methotrexate, because it works on rapidly dividing T cells, and intravenous immunoglobulin, because if you use something to suppress B cells, I'm going to make the child very sick. And the bottom line is, you come up with an algorithm. I'll say that these are my high-risk patients who are CRIM negative. I'm going to put them on this immune tolerance protocol, and I'm going to see if they don't make antibody titers. So this is the summary, and these are cases also from India. The idea is that these are patients who are started now at 2.5 months. We've now instituted the immune modulation at the time of enzyme treatment, and let's see how these patients have done. These patients now, when you look at their peak titers, the median peak titer is 200, and the last titer on ERT is only 6400. It's usually 0. And for a CRIM negative patient, it's normally not 200, it's not 400, it's not 12,000, it's in the hundreds of thousands. So now I'm showing that I'm not making antibody titers. How is it impacting the patient? This is the left ventricular mass, because they have massive cardiomegaly. So this is a group of children who are CRIM negative, who are only treated now with enzyme treatment. You can see that the heart comes down and then starts to go up. This is because of the antibody production, so the enzyme is not having its impact. And now when I use this other cohort of patients in black, and if I pre-treat them with immune modulation, you can see that they come all the way down and stay down. How does that matter? Do they live longer? Look at the antibody titers. This is in the group of patients who are only treated with enzyme therapy. You can see the antibody titers keep going up, versus the group of patients who are treated with immune modulation. And so how does this ultimately impact you? I'm going to bring you back full circle to baby John, and this was him at 9.5 months. And this is now another baby who's CRIM negative, who has been treated with immune modulation, and you can see is walking and doing well. And then here is our oldest little girl, treated, who was CRIM negative, from Israel. And this is her. She received immune modulation in the naive setting, and you can appreciate that she is today alive at 9 years of age. So now let's go to the next concept. We've developed treatment for Pompe. I'm not happy because it's not completely treating these patients. So can I do something else and beyond? So the idea is, remember, going back to basics, I'm dependent on the mannose-6-phosphate receptor to bring my enzyme to the lysosome. But if my concept is that I don't have enough of the mannose-6-phosphate receptor, I'm not going to be able to do this in perfection. So we showed this in collaboration with Dr. Nina Rabin, that the muscles of mice, and also in patients, they have low mannose-6-phosphate receptor expression. And so if we did a strategy where we could increase the amount of mannose-6-phosphate receptor, could we improve the enzyme delivery to these patients? And so without making much ado, this is the normal pathway, again to show you how the enzyme ends up in the lysosome, and some of it is through the endocytic pathway and some of it is through the secretory pathway. And this green is the M6P receptor, which also gets recycled. So now if my concept is that here is a patient now with the enzyme deficiency with Pompe, I'm really dependent on this secretory pathway for making the enzyme available to end up in the lysosome. So now the concept is getting clearer. If I'm able to enhance this pathway by increasing the number of M6P receptors, all these little green figures here, then the enzyme that I'm giving is going to have a better chance of getting caught. And if it has a better chance of getting caught, there's a better chance of its delivery. So we started this now in terms of a proof of concept with a very simple agent, albuterol, or a beta-2 agonist, and now with clenbuterol, which we know is available really in the horse, in the animal world, to make muscles hypertrophy. So for these trade horses, etc., that's what's used. And the idea was, could we increase this myozyme receptor or the M6P receptor? And we were able to show this next in clinical trials in patients with Pompe disease as a proof of concept that patients who were not showing any further benefit on enzyme therapy alone were now showing an enhanced response with the addition of albuterol. So as you can see here, we showed it by the 6-minute walking test, which is again just a point to make that you've got to find a way to show whether a therapy works. And here is the proof of the pudding, so to say, is this is the M6P receptor before and after. And you can see that there's increased receptor after the addition of the albuterol in these patients. And here is the before and after in terms of a muscle biopsy. But of course, I want to make it clear that this is just proof of concept, but the field keeps going on. So we've done this for Pompe, but can this be used for mucopolysaccharidosis type 1 or Fabry? Absolutely. It can be done for the other conditions which are dependent on the M6P receptor concept. And so in summary, the lessons learned from Pompe can shed light on other disorders, and the lessons from Pompe are important for other neuromuscular disorders. It's the first disorder where the pediatric trials paved the way. We have newer therapies on the horizon. The beta-2 agonist is a Duke approach. Genzyme is working on a second-generation enzyme. BioMarin is working on a second-generation, as is Amicus. But I would like to say that the importance of collaboration and bench to bedside and bedside back to bench is the key to your success. And working together as a team is another key to your success. And so this is the concept that I have really lived by, is we took work from the bench, took it to the bedside, and we learned from the patients, took it back to the bench, and we worked in this circuitous way, not just for Pompe, but for other rare diseases. And so this is something that I put together for myself over the years of how we developed this treatment. Each year was an added concept, and each year we kept learning more. Each year a patient added to my experience, and then this is what brings me the most happiness, is that we were able to change the natural course of this disease, but yet we're not completely satisfied, which then took us to the concept of newborn screening, which today is approved, and I think I'll talk about this in my next talk, that if I can start very early. And so I want to tell you how I've taken the concept from Pompe to another rare disease where I've been a part of the story of hypophosphatasia. And remember, for Pompe, we talked about the mannose-6-phosphate receptor being very important, so the addition of the sugar moiety to get it to the M6PR. Now for hypophosphatasia, which is a rare skeletal dysplasia or a bone disease, you need to target the enzyme to the bone. If you don't target to the bone, it's going to be lost. And so this was a concept that was developed by Enobia, and then the conduct of the natural history studies and the phase 1 clinical work all started again in collaboration with us at Duke. So I'm just going to keep this concept. Again, it's an ultra-rare progressive genetic disorder caused by mutations in the alkaline phosphatase gene. And the way you diagnose it is you look for low levels of alkaline phosphatase, ALP. Very often it's low, and I guarantee you, I know I have missed cases because in my institution, only high values are flagged and not the low values. But that's another point to make. And so this disease includes severe demineralization and death. because of respiratory compromise in infants and crippling fractures and bone pain in adults. And up to recently, there was no approved treatment. So again, it's a continuum of disease. One hand, you have the perinatal form, which is lethal, and then you have the infantile form, childhood, adult, and then primarily with early loss or premature loss of teeth. And so this concept is again that if you don't have this particular enzyme, you're going to have the clinical sequelae that impact the skeletal and other major organ systems. And so it's got significant mortality, nearly 100% of those who manifest at birth, and approximately a 50% mortality in patients who manifest shortly after birth. So keep this in mind, another rare disease, and can we do something to impact this disease? The story is repeating itself. Now a natural history study showing you that it's almost 100% lethal. This is used as your comparator group, and now you do the understanding of this bone disease. These are what we call the radiolucent tongues, and here you can look, there's really no bone, and this is a child at 36 months of age, and this was that child at 7 weeks of age. It's the same child, by the way. You can see over a period of time, there's a complete undermineralization of the bone. And this is now the skull also showing absolute hypomineralization and this copper-beaten appearance. And the idea is now if you do an enzyme replacement therapy, but you use the bone targeting, so you use the FC fragment and you use this asparagine tail, you can target it to bone. And I'm going to show you some very exciting videos. This is at baseline of a child, and you can see how difficult it is for this baby to breathe. This would have been a child who would have died. This is 6 months into treatment, and this is 18 months into treatment. And this product today, aspartase alpha, just received FDA approval less than a month ago in the US, but already has approval in the European Union and also in Canada and in Japan. And here is another patient with the disease, and you can see after 6 months of treatment and then after 18 months of treatment. So you can apply the lessons from one rare disease to another, apply these concepts of targeting and make a difference. And so I'm going to show you the impressive improvement in bone. So this is at baseline, and you can definitely appreciate the undermineralization and deformed bone. You can see that here there's radiolucent tongues and there's the appearance of rickets. This is at week 24, and then this is at week 48. So remarkable improvement in terms of mineralization. And here, if I'm running out of time, should I just stop right here and just talk about these concepts, or? I can? Okay. So there are now other treatment approaches. We went from simple to replacing the missing enzyme, and now we can think about substrate reduction therapy, chaperone therapy, read-through therapy for nonsense mutations, and other therapies. And I'm just going to do one slide for each one of them. So the concept of substrate reduction therapy, if you remember, we talked about a product, an end product, and an enzyme. So if you're missing the enzyme, you're going to build up that substrate. So if you can reduce the accumulation of the metabolites by inhibiting their synthesis altogether, it can be very, very helpful. So here we have the example of Gaucher disease, for which we have a treatment. We know that the missing enzyme is glucosylceramidase or glucocerebrosidase, and the product which accumulates is glucosylceramide. So here, if we can prevent by this process here, the glucoceramide synthase, then we have less of accumulation of the substrate. And if you have less accumulation of the substrate, maybe the body's residual enzyme can do its job. So this is a concept which again is an approved therapy today, and I think Dr. Balodi is going to speak about this. And you can see that there's remarkable improvement with this particular therapy. Improvement in the spleen and liver volumes, improvement in hemoglobin, improvement in platelets, and also improvements in the spine density. The second is the concept of bone marrow transplant, which we know is very successful. The third is the use of chaperone. And what is a chaperone doing? A chaperone is really binding and stabilizing the misfolding prone proteins. So some of our proteins are in the body, but they are misfolded. And if they are misfolded, they can't go to the target sites. So take this concept, and now if you think about this, this is the enzyme, and this particular enzyme, if you had a chaperone, which is in yellow here, it can stabilize the enzyme, and then if it stabilizes the enzyme, it can allow it to go to its target site, which in this case is the lysosome, and then that substrate or the chaperone has to retract itself and allow the material that was not being digested to be cleared. So this is the concept of chaperone, and can be done along with enzyme replacement therapy as a combination approach. The other one is the intrathecal approach to enzyme, because we know that enzyme therapy cannot cross the blood-brain barrier. So that's another concept which is in clinical trials. And then the idea of combining therapies, which we saw, enzyme replacement therapy plus small molecule therapy. And this is really the current but the exciting time that we are in, Duchenne muscular dystrophy. Majority of these patients have deletions, but there is some patients who have these premature stop codons, and nonsense mutations are seen in many of these genetic disorders, and they create this premature stop signal in the translation of the genetic code contained in the mRNA. So if this happens, you're not going to have the production of the full-length functional protein. And an example of this is a small molecule, atalurin, which has the therapeutic potential for Duchenne, for cystic fibrosis, and for other genetic disorders. And the idea is here. This is normal translation, and you've got the normal stop codon and the normal stop signal here. But if you've got incomplete translation, because you've got a premature stop codon, you're not really completing translation, and so you've now got a much shorter truncated protein. But now if I provided a small molecule which would allow this read-through and allow it to go to the correct stop codon, we can really have another treatment. So this atalurin actually does have some kind of approval in the European Union right now for Duchenne muscular dystrophy. And then we have other concepts, antisense therapy, where you have a small sequence of DNA or RNA, which is termed an oligo, and the goal is to alter the specific gene expression. So I'm going to close by talking to you about human gene therapy, because I know this is something that Dr. Phadke wanted me to speak about. There are many disease targets, ranging all the way from AIDS to our genetic disorders, and there are many different clinical trials. So if you were to do a clinicaltrials.gov search, you'll find a whole slew: cancer diseases, monogenic diseases, cardiovascular diseases, gene marking, healthy volunteer studies in terms of gene therapy clinical trials. And I think you've got to consider what kind of gene therapy you're doing, whether it's somatic or whether it's germline gene therapy, and that gene therapy at this point is really limited to somatic cells and disorders that are caused by a single gene. That's where the field is right now. Doing germline gene therapy is currently banned. And so the idea then is you can do it ex vivo, that's outside the body where you manipulate, or you do it in vivo, where the gene of interest is introduced directly into the specific cells of the body. And this is the one that's more commonly done right now, the in vivo approach. So which conditions would I like for gene therapy? It needs to be really a lethal disorder where the course is not very variable, which can be reversible. We don't have an approved or a good therapy. The gene has been, of course, cloned, otherwise how are you going to do gene therapy? No tissue specificity or regulation, and bone marrow cells, if they're involved, is an advantage. So you can use different kind of vectors for this, ranging from retrovirus, adenovirus, AAV vectors, herpes simplex, liposomes, naked DNA, plasmid DNA. This is a whole talk in itself. But to tell you that the field of gene therapy is vast, completely vast, and there are different approaches that can be used. So once again, to show you, different kind of vectors are being used in various different gene therapy clinical trials. And I think I put this in parentheses, successful gene therapy in immune deficiency diseases, which we heard about in 2005 for X-linked SCID. And then we know that there was that oncogenic insertion in 2 children who then went on to develop leukemia, and that put a huge halt to the progress of gene therapy trials. But now we're coming back full circle and new trials are underway. And then there is approval in our rare disease space, a gene therapy for lipoprotein lipase deficiency. It's called Glybera. It's a one-time injection. It's approved in the European Union. The company is called Unicure, and it's using the AAV vector, or the adeno-associated virus.
Sorry to
and
interrupt.
the goal right now is to move to other diseases like Gaucher disease, Pompe disease, and other conditions, glycogen storage disease type 1.
Sorry.
But I'm going to leave you.
Yeah, we just stop now. I think
Yes, I'm done. What do you really think? Think about the Jesse Gelsinger story. We still have to be very careful in the field of gene therapy. And here you can see the field has expanded. These are all in the last 3 years, newly approved therapies by the FDA for the rare diseases. I'm not talking of other diseases. And therapies in development. If you can take a moment to see the technologies, RNAi, substrate reduction, protein-based therapies, enzyme therapies, nonsense read-through, alternative splicing. So some of the concepts that I've mentioned to you are currently in clinical trials. So for any of you, please get interested in genetics because the field is wide open. Our field is exploding. There are different approaches which I've just whetted your appetite here. But the most important is to work together and to think outside the box, listen very carefully to your patients and follow your passion. And I really want to thank the committee, Dr. Kuldeep Singh, Dr. J.P. Soni, all my colleagues and the faculty and staff at AIIMS Jodhpur, my patients, and I really have to take a moment today to dedicate this lecture to my dad who just passed 2 weeks ago, who inspired me in this field of genetics and to pursue my passion. He's the one who sent me to the US. He's the one who encouraged me every step of the way. Thank you.
Thank you, ma'am. Thank you, Chairperson. May I request Dr. Ashwin Dalal and Dr. Shubha Fatke to present the award to Dr. Priya Krishnani. Thank you, ma'am. Thank you, sir. May I request Dr. Ashwin Dalal to present mementos to our chairperson, Dr. Dhavendra Kumar, Dr. Anurag Singh, Dr. Pramod Sharma. Thank you, sir. It's time to begin third session of today's day 1 of the conference. It's on diagnostic avenues, and the chairpersons for the session are Dr. Manish Parekh, Professor, Department of Pediatric Medicine and Consultant Pediatric Neurologist, Dr. S.N. Medical College, Jodhpur, Dr. Suresh Verma, Associate Professor, Department of Pediatrics, Dr. S.N. Medical College, and Dr. Pratibha Singh, Additional Professor and Head, Department of Obs and Gynecology, AIIMS Jodhpur. May I request the chairpersons to take their seats and initiate the session.